Hello everyone, welcome to Agri Question and Answers. Here we'll see some bits in fundamentals of genetics. Firstly, the biological science that deals with the mechanism of heredity and the cause of variations in living beings is called as genetics. The term genetics was derived from the Greek language. Gens means to grow, and the term was given by Bateson. Father of genetics is Gregor Mendel. Next, chromosomes are longest and thinnest during interphase. Chromosomes are smallest and thickest during mitotic metaphase. Among the higher plants, the longest mitotic chromosome are those of Trillium species. Centromere consists of two disc-shaped bodies are called as kinetochores. Two ends of chromosomes are known as telomeres. The differential staining capacity of different parts of a chromosome is known as heteropyknosis. Arranging the chromosomes in the descending order of size, keeping their centromeres in the same line is called as karyotype, which means the characteristic feature by which the set of chromosomes of a species. The karyotype of a species can be represented by diagrammatically, and showing all the morphological features of chromosomes is called as ideogram or idiotype. The term mitosis was coined by Fleming, whereas meiosis J.B. Farmer. The chromosome number is constant, but the quantity of each chromosome reduced to half in anaphase of mitosis. The first division of meiosis results in the reduction of chromosome number to half is called as reduction division. In zygote, the pairing of homologous chromosomes from bivalence are called as synapsis. The centromere of the bivalents are arranged on either side of the equator, and this type of orientation is called as co-orientation. External appearance of an individual is called as phenotype. The genetic constitution of an individual is called as genotype. Progeny obtained by the crossing of two genetically dissimilar parents is called as hybrid. The cross between the F1 hybrid and either of its parents is called as backcross. The cross between the F1 hybrid and its homologous recessive parents is called as test cross. The manner in which genes control the phenotypic expression of various characters in an organism is known as gene action. Interaction of genes at different loci that affect the same character is called as epistasis. Degree of phenotypic expression of a gene in different individuals are called as expressibility. Some genes are known to affect or control more than one character is called as pleiotropism. The ability of a gene to express itself in all individuals which carry it in the appropriate genotype is called as penetrance. Tendency of gene to remain together in the original combination during inheritance is called as linkage. The most common method for detecting the linkage is called as test cross. Sum total of genes present in the cytoplasm of cell or an individual is called as Plasmon. Nucleotide consists of sugar, nitrogen base and phosphate group, whereas in nucleoside sugar and nitrogen base is there. Here adenine guanine equals to cytosine and thymine. This is the Chargaff law, which means purines equals to pyrimidines. DNA replication always proceed in the direction of 5' to 3'. In RNA, thymine is replaced by uracil. In tobacco mosaic virus having genetic material is single-stranded RNA. In tRNA is also called as soluble RNA. The process of production of mRNA from the DNA is called as transcription. The process of protein synthesis from the RNA is called as translation. The initiation codon is AUG. AUG code for amino acid is methionine. Triplet code first suggested by Gamow. Wobble hypothesis was proposed by Crick. Lac operon in E. coli was proposed by Jacob and Monod. The sudden heritable change in the character of an organism, which is not due to either segregation or recombination, is called as mutation. A physical or chemical agent that induces mutation is called as mutagen. A mutation which kills all the individuals that carries it is called as lethal mutation. Induction of mutations in bacteriophage is by proflavin. A single base in DNA molecule is replaced by another base is called as base substitution. Replacement of purine by another purine or pyrimidine by another pyrimidine is called as transition. Replacement of purine by pyrimidine or vice versa is called as transversion. Loss of segments of chromosomes is called as deletion. A chromosome segment that contains genes in a sequence which is reverse of normal is called as inversion. Thank you everyone. Subscribe Agri Question and Answers.